what a great event to be here in Seattle with talking about science, talking about Nobel Prize winners, talking about what really counts. I want to talk to you a little bit about Mary and what she's done. And for those of you that are science nerds, she identified a gene called the FOXP3 gene. And it really ultimately shows how the body protects itself from attacking healthy tissue, transforming and understanding the immune system, and advancing new treatments, as I said, for things like autoimmune diseases, lupus and rheumatoid arthritis, and transplant patients. That's made a fundamental impact that all researchers use today. And the breakthroughs are really what families were hoping for. And the thing I need to emphasize is science takes a long time. And when you cut it off, we're not going to see all the cuts in funding that we've seen today. We'll see the effects of that 10 years from now. And it is one of the things I would say that's made the United States great, has been our innovation, our love of science and what we do. And to see that destroyed has been very, very painful for a lot of us. I think Mary's work shows what great science and great research can be about and the effects that it can have on for society. She currently is still a distinguished investigator at ISB and continues to lead interdisciplinary research at what we call the intersection of genetics and computational biology, which our colleague, Dr. Lee Hood, was talking about probably 20 years ago. Joining Mary on the stage will be Katie Hafner. Katie is an acclaimed journalist, author, and the co-founder and host of Lost Women of Science, a podcast. Her work focuses on illuminating the remarkable lives and legacies of female scientists whose contributions have been overlooked through history. She's a veteran of the New York Times, and get a load of this diversity. She's the author of 6 nonfiction works exploring diverse frontiers, the origins of the internet, the world of computer hackers, German reunification, and guess what? The life of Glenn Gould, the pianist. So if you can put those things together, that you got to have someone who's clearly exceptional. Tonight, Katie and Mary will explore the landscape of women's leadership in science, alternative pathways into STEM, and the critical mission of mentoring and supporting the next generation of scientific pioneers. Let's welcome both of them with a round of applause.
That was nice.
That was super nice.
Thank you, Rod. And I will thank Jim Heath, Rod Hochman, and Lee Hood for tonight and all of tonight's sponsors. And let's just get into it. Did you want to thank someone?
I also want to thank Jim Heath and
ahead.
Lee Hood and Mike Collins and the whole team that's put together this fabulous event. And I really want to thank my family for the support. My husband, Ross, Lauren, Devin. Has been incredible.
So let's start with something really light and fun.
Okay.
Then we'll get into the heavy stuff. Okay. So the story of the knock at the door. I know a lot of people have heard it, but it's one of those stories that just sort of, probably gets better in the retelling. So tell us the story of the knock at the door.
The knock at the door. This was October 6th. The knock at the door was preceded by a couple of phone calls that I ignored.
In the middle of the night.
In the middle of the night at 1 a.m. But at 3:15 a.m., there was a knock at the door, and my husband wasn't going to let her in. There was a young woman on our porch with a couple of big cameras slung around her neck, AP photographer. She convinced Ross that, there's a Nobel Prize winner in this house, and I was told to come take a picture. So that was the knock on the door. And then once I woke up, realized that, oh my, there's a whole bunch of voicemails and text messages and emails and all kinds of stuff that I missed at 1 a.m.
So these calls were coming in and you were just like, oh.
Yeah, no. Yeah, I mean, who takes a call from Sweden in the middle of the night?
Yeah, who does that?
Well, I will next time. And I really recommend, if this happens to anyone else, that you just answer the phone. Yeah.
So you were completely surprised by this, like really bowled over,
Yeah.
and you did not even know you'd been nominated,
Correct.
and which was unusual, as I understand it. I mean,
Yep.
okay, well, let me just start by saying that, and this comes from somebody named Gustav Karlstrand, who's a Nobel Prize historian. And when the Nobel Prize was started in the early 1900s, because it was kind of this old boys network and everybody knew each other, there was this rule that it had to stay confidential, that you could not say who had been the nominator. And then in the 1970s, the rules changed so that you can know, but you have to wait 50 years. So,
Bummer. Yeah.
so, and yet you didn't know. And then I think there was someone who was surprised that you didn't know.
There was somebody who was surprised, who was like a documentarian of all Nobel Prize winners, and he came to Seattle to interview and take pictures. Peter Badge couldn't believe that I didn't at least have an inkling, and I don't know why that is, because it is supposed to be confidential.
Right. I used to cover Apple Computer way back when Steve Jobs was there. And he used to say, "Isn't it funny how news leaks from the top?" So who knows how word spreads? You get the news that you've won, and there's a lag time between when you did the work and made the breakthrough and you won the prize. And I think there are some famous examples. It's an interesting bit of Nobel Prize history that actually the lag time through history has increased, which surprised me when I was looking into it. So in the early 1900s, there used to be something like 5 to 10 years between when the breakthrough was made and when the prize was won. And now the average is more like 20 to 30 years. So you're right on schedule. So let's talk a little bit about the science, if you don't mind. And I know a lot of people know and understand the science, but for those who don't, can you give us a little primer?
Yeah, so when I was working at Darwin Molecular and Fred was there as well, I was there because of my expertise and experience in genomics and genetics and molecular biology. And we decided to use this mutant mouse called Scurfy that had an interesting autoimmune phenotype. And the idea was, if you could understand the molecular basis of that phenotype, maybe that would give you an interesting target for drug development. And so we cloned the gene. These mice have a really serious autoimmune defect that causes really massive lymphoproliferation. And so it seemed like maybe this would be something that is pointing to a really important regulator of the immune system. And indeed, that's what we found when we cloned the FOXP3 gene. Then we discovered that it's a very good marker of what is known as regulatory T cells, which are really critical for sort of maintaining homeostasis in the immune system. So making sure that the immune system doesn't attack self-antigen, but still allows the immune system to attack the things that it's supposed to, like viruses and bacteria and whatnot. So it's a really critical way of keeping the immune system in balance, homeostasis, and it's peripheral immune tolerance is the term that we use.
Later, we're going to talk a little bit about history and the build up to this, but I wanted to ask you, when you started out, my guess is that you weren't setting out to make this discovery. It's like, oh, I'm going to discover something that's going to like be really super significant about the immune system. So tell me, when you were starting the work, was it just curiosity? Was it an open mind? Tell me what it was.
It was, well, for me, it was just an excellent opportunity to kind of demonstrate the power of mouse genetics, for one thing. So, starting with a really interesting phenotype that has clinical relevance and showing that you could identify the underlying basis of that phenotype. And I was not an immunologist, so in fact, the description of the mice themselves and the defect in them was, a little bit mysterious to me, but I could understand the genetics and the molecular biology and could tackle it from that point of view. And I knew that, just given how severe this phenotype is and what the mice were teaching us about the importance of keeping a damper on the immune system, I knew it was going to be something, exciting and critical, but of course could never have predicted that it would, really change the way that immunologists think about the whole system.
Which brings me to this whole idea of what happens through history, what you built on, and what came before, and what you've said about sort of the time is ripe when the timing is good. So let's go a little bit back in history. So we're going all the way back to Oak Ridge, right? Tell us about the mice.
Tell us about the mice. So Oak Ridge, so the story of the scurfy mouse goes back 75 years or so. So
Amazing.
yeah. So back in the mid 40s, so during World War II, as part of the Manhattan Project, the Oak Ridge National Laboratory was set up, and for the most part, that facility was there to help develop the atomic bomb. But they also had a real recognition for the importance of understanding what the effects of radiation exposure are. So they also set up a mammalian genetics unit, and they brought in a couple of really top-notch mouse geneticists, Bill and Leanne Russell, to oversee these studies to look at radiation exposure. And really, much of what we know today stems from those studies. And they, being mouse geneticists, they really benefited from the program because in the end, they ended up with this amazing collection of mouse mutants, which have been a huge boom to the scientific community and have taught us a lot about all different biological systems. So people have been studying these mutant mice for a long time.
So they got the mouse ball rolling.
They got the mouse ball rolling.
And then what happened in 1960? There was a Nobel Prize. So I'm trying to build this whole scenario of how this doesn't happen in isolation, right?
That's right. Every new discovery is built on something in the past. And so the idea of immune tolerance was first described and honored by a Nobel Prize in 1960. So the concept of there being a way to make sure that the immune system doesn't recognize self and destroy, your own tissues was not a new concept. But the peripheral immune tolerance that these regulatory T cells represent was new.
Right. And so talk to me about the whole idea of what I call the time is ripe phenomenon. Like when you were working on this, it was the right time scientifically, but also sort of from a collaborative point of view, right? Because you've talked about the importance not just of the science, but of communication. So talk a little bit about that.
That's right. There in a lot of ways, the success of this project had to do with a lot of things coming together at the right time. And a lot of it, I think, was the right set of people. So the team that I worked with at Darwin Molecular was amazing. It had just the right mix of, a diverse set of expertise and experience and a real sense of common goals and common drive. But equally important, it was a time when the tools of genomics were ripe and ready to be used in this way. So the Human Genome Project was underway. there were these new technologies. Lee Hood actually had a huge hand in developing things like automated sequencing that made it all possible to do the kinds of kind of big DNA experiments that we needed to do. So we couldn't have cloned the gene, 5 years earlier than we did because we just didn't have the tools. And then the equally important was the people that were put together to do the job.
Right. And one of the things, as Rod mentioned, I'm an internet historian, and there's a man named Paul Barron who invented packet switching, which is kind of the technical underpinnings of first the ARPANET and then the internet that came after it. And I wrote his obit for the New York Times, and I pulled out this quote that I really wanted to share with you about it's sort of the standing on shoulders idea. And what Paul said was, and he was a very humble guy, the process of technological development is like building a cathedral. Over the course of several hundred years, new people come along and each lays down a block on top of the old foundations, each saying, I built a cathedral. Next month, another block is placed atop the previous one. Then comes along a historian who asks, well, who built the cathedral? Peter added some stones here, and Paul added a few more. If you're not careful, you can con yourself into believing that you did the most important part. But the reality is that each contribution has to follow onto previous work. Everything is tied to everything else. And I think everyone in this room gets that. And I wanted to ask you, so this goes completely counter to what we at Lost Women of Science find a lot, which is through history, what we call the great man theory of scientific discovery. Do you believe that the great man theory of scientific discovery is a thing of the past?
I don't know if it's a thing of the past. I think we've moved beyond that, and there's a great understanding that the best way to get things done is to do it as a team, especially in these days where the science is so complicated. It's very hard for one person to be able to be an expert in everything, and it's best to pull together the best team.
And you also say something interesting about being in an academic institution versus where you work. Talk to me a little bit about that.
Yeah, I really felt, I've never had a position in an academic department where I was leading my own team, but I feel like you can really make a distinction between that scenario and what things were like at Darwin Molecular. And again, this is an N of one for my company experience too, working at Darwin. But I think Darwin was unique in that everybody really had a common goal. And there was there weren't silos that separated groups. Everybody really worked together because it really was in the best interest of everyone to complete the goal. Whereas in academia, I think there's more, you really need to take care of yourself also. There's so much pressure to get your own grants and write your own papers, which is not a bad thing, but it's just a different way of approaching things, I think. So that was my experience, is that, and I felt like I really thrived in that experience where, there was real kind of equality. There was really not much of a hierarchy within Darwin, and we all knew what needed to be done in the end.
However, you were in academia when you had your two main mentors. Talk to us about the importance of mentorship in general and mentorship by a woman in particular.
Right. So yes, I would count two. I've had several really meaningful mentors throughout my career, but I would say the ones who have had the greatest impact on me, and certainly the one who made me go into research rather than medical school, which really, there's no way I'd be sitting up here as a Nobel laureate if I hadn't gone into a lab. And that was Dr. Larry Sandler at the University of Washington, where I was an undergrad and I did a research project in his lab and just totally got hooked on research, which had not been my plan. And he was an amazing mentor and teacher. For my PhD thesis, I was at Princeton University in the lab of Shirley Tillman, and she really is a giant amongst the women in science. And she has done so much for promoting young women, but really anybody in her orbit is lucky to be around her. Super smart, rigorous scientist, but also a real champion and really takes great pride in teaching her people, how to think critically and communicate well. Yeah, I would say as a mentor, she was critical, and as a woman mentor, it was really important to see her as a role model and to see what was possible.
Because she had kids at the time, right?
Yes, she did.
Yeah.
And single, she was raising the kids on her own and running this lab, and yeah, she was amazing what she accomplished.
Very much the proverbial backwards and in high heels. Yeah. So, on this note of women in science and not just carrying the torch, but passing it forward, I wanted to invite onto the stage a couple of young women to talk about their experiences so far. They haven't lived long enough to be old and wise, but they've sure gotten a great start. And they are Layla Ismail, who's a former ISB high school intern. Layla is currently an undergraduate student at Harvard. And Crystal Perez, who went to Stanford and is a graduate student at UW, working in ISB's Gibbons Lab. I'm asking the first question to Layla. Layla, you first came to ISB as a high school student. Was there a moment during your internship when science stopped being something you learned about in class and became something you could imagine yourself doing?
Yeah, hi everyone. So during my time at ISB as a high school intern, and I got the opportunity to interview about 15 different scientists. And throughout these conversations, I began to learn more about these different scientists, their paths to research, their passions for science, and most of all, their background. Coming from somebody who went to underfunded school in South Seattle, the first person in my family to go to college, science always felt like this far off dream to me. And I think throughout these conversations and talking with these different researchers, I really began to put myself in their shoes. I remember the first day of my internship like it was yesterday. I was filled with such an imposter syndrome because I didn't have that traditional wet lab experience and background. And honestly, I think throughout these conversations that I had with all of these different people, it really increased my self-assuredness and my confidence, which has allowed me, I think, to apply to Harvard, go to Harvard, and even do research at different R1 universities across the United States.
And then Sierra.
And I get to ask you a question as well. Looking back, Layla, how did your experience at ISB influence the educational path you're on today? And what did you learn about yourself during that time?
Yeah, great question also. So as I said before, I think ISB really gave me the tools to start imagining what I wanted for myself in my future. I remember in high school, my biology teacher recommended that I apply to the internship because he knew I was very generally interested in science. And then I think back to at the end of my internship, I became so enamored with cancer biology and all of these things that I very advantageously decided that I wanted to pursue an MD PhD. And I remember even at the end of that experience, really tearfully hugging my mentor and thanking her for the opportunity and experience that she gave me. I think after those 6 weeks, I had a clearer picture of the life that I wanted to lead, one that was full of science, curiosity, research, and all these investigative questions. And I think I can really credit that to ISB because it showed me a world of opportunities and learning that I had previously not had the opportunity to look at. And then I think most importantly, what I had learned was how to push myself and be in rooms where even initially I feel nervous. I think that when youth from disadvantaged backgrounds who are minorities or women are given opportunities like this, they leave from these places with a huge amount of confidence that is instilled within them. And I think that we should increase opportunities like the ISB internship and places like ISB all across the United States. And yeah, just replicate what I was able to gratefully, very gratefully be a part of.
And then, Crystal, I get to ask you the first question, which is, as a graduate student working in a world-class research environment, what has surprised you most about how science actually gets done?
I think people have already touched on this a little bit, but probably how slow and iterative it is. And I think I've always known that it's going to be slow, but I have found myself, for some context, I'm about halfway through my MD PhD program, 2 years into my PhD. And for the first 2 years, I found myself learning new things I didn't expect from my experiments. And then these new things lead me to ask new questions. And then I have to ask for help from collaborators. And so my first project is about the effects of recent and not concurrent antibiotic use on the gut microbiome and metabolome. So it started off with 2 small questions, microbiome, metabolome. And now I have 2 sets of collaborators, internal at ISB, some at Albert Einstein and Fred Hutch. And so it's been 2 years, but it's been awesome to see it grow and snowball into something.
Okay, before Mary asks her question, I want to interject with this whole, what you just said about how many people you're involved. So when you win yours, take a page from Mary. I mean, both Mary and Fred, the number of people they thanked in their Nobel lectures. I mean, Fred thanked the mice. So I just want to say thank you for mentioning just how many people are in your orbit.
That's so important. So I have a question too. We've talked a lot about mentorship and scientific lineage. What has mentorship meant to you during your training, and how has it shaped the scientist you are becoming?
Yeah, I think this is a great question. And in particular for me, I think that doing my PhD at the ISB, and in particular the Gibbons Lab, it was exactly what I needed. For starters, Sean Gibbons, who's my faculty advisor, he's exactly the kind of investigator that I want to grow to be one day. He has such a strong work ethic. He's curious, the sheer volume that he reads, science, non-science. But then around me, I have older graduate students and postdocs, young faculty who are great near-peer mentors that I can emulate. And so, yeah, as I mentioned, I'm around halfway through. I just had a committee meeting to discuss taking my candidacy exam in the fall. And just like looking back and thinking, it's awesome to feel myself grow. My faculty committee, my mentors, they can ask me questions. And I feel like I can provide insightful answers or explain my approach. And so now I feel like I'm becoming their peer in science. And that's really awesome, but I couldn't have done it without them.
I love that. I think we have time for very brief, because I really want to hear this from you guys. If you were going to, the two of you, that is, if you were going to give a piece of advice to someone who is a young person who's curious about science, but they're really not sure whether they belong, what would you say?
I would say to keep reading. As you guys can see, I'm such an advocate. Keep reading and keep talking to new people, because you might find some areas of science where 2 things connect that you might not have expected. It might drive you to keep going. You might find a corner of science where the people are just like exactly what you need, but you won't know until you keep exploring.
Yeah, my piece of advice I think would to just be very comfortable with being uncomfortable, be able to put themselves in scenarios and rooms and challenge themselves every day with new opportunities that I think they would have, as I did, felt very nervous about beforehand. Yeah.
That's great. Thank you. So thank you so much to both of you. And Mary, oh my goodness, Thank you so much for doing this. I mean, for doing everything.